So today I'm teaching Shelby and my cat Bonnie about DNA, RNA, and protein synthesis. So to begin with, Shelby, protein synthesis occurs in the cell cycle, and it occurs in the G1 and the G2 phase. So the G1 phase is called the interphase, and in the G2 phase, that's basically where the DNA is made. So a little background of DNA. It's made up of 4 different things, and A binds to T, which are 2 chemicals, and C binds to G, which makes up the 4 letters of it, A, T, C, and G. Now, RNA is super similar, just like my cat and my other cat here. Come here. You can kind of see him. Both cats, they're both cats, but they don't look alike, though they are similar because they're both cats. So that is basically DNA and RNA. As I was saying, DNA and RNA are super similar. The only difference between DNA and mRNA, which the M in front of the RNA stands for messenger, so it's a messenger RNA. So as I was saying, how DNA has A binding to T and C binding to G, in RNA, instead of a T, there's a U, so it's A binds to U and C binds to G, which is basically the only difference. So now I'm going to move on to talk about transcription and translation. So transcription is the making and the copying of the DNA. So basically, if you think about it, DNA is like a zipper. When you need copies of the DNA, that zipper opens up and it splits the 2 sections in half. So in transcription phase, you'll need the one side of the DNA that has the gene to copy it for the mRNA. So once that's copied, then the mRNA, so like I said before, the messenger RNA, then leaves through the nuclear pores and goes into the cytoplasm to find a ribosome. So translation, if you look at it in the whole English language, is basically when you take one language and transfer it into another. So in an easier term, if somebody's speaking French to you, then you can then go online and translate it into English to understand what's going on. So it's the same type deal for translation in the whole proteins, high five, in the whole protein synthesis thing here, is that mRNA is read in 3 groups called a codon. So the starting codon for the whole mRNA is always AUG. So when we get a long strand of the mRNAs together, it can be something like AAUCG, but until you find AUG together, which is the start codon, you can't start the process. So once you find the codon AUG, Bonnie, stay still. So once you find the starting codon AUG, that's the start of the whole mRNA and chain. mRNA. So then you get something that's called tRNA, so it's basically your transfer RNA. Oh no! So basically, as I was saying, you get something that comes along called the tRNA, which is your transfer RNA. It comes along and it makes the anticodon, so the opposite to the codon, AUG, or whatever's following the AUG start codon. It puts the opposite besides it, or below it, I should say, actually. And then it also brings in the amino acids to form a protein chain. So that's basically translation in a nutshell. So there are actually 3 types of RNA. So the mRNA, which we discussed, is the messenger RNA. There's the tRNA, which is the transfer RNA, and there's also the rRNA, which is kind of a ribosomal thing. So now that we've talked about the different types of RNA briefly, I'm going to talk about mutations, because everyone's so interested about mutations. So there's actually 2 factors that can cause mutations. So basically the first one, which is the most common, is just a mistake in making your DNA, which there's actually 2 different types. So in the whole mistakes in making your DNA, there's something called a point mutation, which is basically just one of the bases in the whole DNA chain, so the A, T, C, and G. One of those letters can be scrambled, so let's say, for instance, the chain was supposed to read A, T, C, and G. So in this, the point mutation, where the single base of your DNA gets substituted, it would, instead of being ATCG, it could be AACG. So just that one letter gets changed. So it doesn't always alter the chain. It can, but it's very rare occurrence. And an example of that we learned about in class is sickle cell anemia. But then the second type of mutation is called a frameshift mutation, and that's basically where there's either an addition or a complete deletion of the single cell base in your DNA. So for the whole deletion thing, if it was A, T, C, and G, it would have like the T completely annihilated, not in there, so it would be just A, C, and G. Or there could be an extra letter added. Bonnie, you stay still. So it could be AATCG, which then in turn basically changes the whole reading of the frame completely, especially then when you have the RNA beside it, which is trying to match up to the DNA. It's completely messed up. And then it can't read it correctly, and then that's when things such as Down syndrome and whatnot can occur, because the DNA gets to be not read correctly. There's either an addition or deletion, which then in turn you can think that it would mess up something. So, yeah, that definitely changes the whole reading and the whole aspect of it in turn. So the second factor that can cause mutations is environmental. So things like X-rays, UV rays, radiation. Actually, if you think of Chernobyl or whatever, when the nuclear power plant exploded, I do believe, then all that pollution from the radiation was put out into the air, and everyone had to evacuate because it can screw with your DNA. A lot of the people that were alive that stayed got sick, but then the people that ended up staying and living there and whatnot and having babies, their babies were born with a lot of deformities and stuff. Like, I don't want to say, like 4 arms and 6 legs, but pretty severe mutations and whatnot. So it's definitely not healthy to go and get an X-ray when you're pregnant or anything like that. Just not a good idea. Just don't do that. So I'm just going to do a quick recap of everything that I just discussed, just to make sure that everything is clear. So protein synthesis occurs in the cell cycle. It occurs within the G1 and the G2 phases. So the interphase, which is G1, and the G2 is where the DNA is basically split and then made. So moving on to transcription, Kitty, listen up. This is important stuff. So transcription is making a copy of the DNA. So basically, DNA is like a zipper. It unzips down the middle when you need to get things from it. You only need the one side of the DNA to make a copy of the gene. You also then need the mRNA, so M, messenger RNA, which then comes along and binds to and then makes the copy that's identical but different, because as I said, DNA has the 4 different letters A, T, C, and G. A binds to T, C binds to G, and RNA is similar, like my 2 cats that are both cats but look different, are similar but different. And in RNA, it's A binds to U instead of A binds to T, and then C binds to G. So very similar but different. So with transcription, the mRNA comes along, does its thing, and then leads through the nuclear pores into the cytoplasm to find a ribosome. And then with translation, which is basically just how the mRNA is read into a different language, because it's not the same as DNA, you have to have a codon in the chain, and the start codon for the mRNA needs to be AUG. So the chain can be like 20 letters long, but until you find AUG, the chain cannot start. Once you find the AUG, then the tRNA comes along, and it makes an anticodon. And the anticodon then matches up to the actual codon that the mRNA has made, and then the tRNA brings in amino acids, which then forms a protein chain.
Okay.
So, do you have any questions so far?
Can you show me an example of the tRNAs and the mRNAs?
With an example.
Of biology can be summarized by DNA codes for RNA, which codes for protein. mRNA, rRNA, and tRNA are all vital to this dogma and protein synthesis. Though functionally diverse, all three are composed of single-stranded RNA. Protein synthesis begins in the nucleus of the cell. Because DNA cannot fit through nuclear pores, it must be unwound and copied into strands of RNA, which exit the nucleus. These strands of RNA are called mRNA, or messenger RNA. Messenger RNA travels from the nucleus to a corresponding ribosome, where it will be translated and a protein will begin forming. mRNA serves as a messenger for the protein code from the nucleus to the ribosome. As mRNA enters the ribosome, the complex physically moves along the mRNA strand. The ribosome, which is about 60% rRNA and 40% protein, identifies codons and binds a corresponding tRNA molecule. A codon is a sequence of 3 nucleotides that code for a single amino acid. Each codon along the mRNA strand is read by the ribosome and matched up to a corresponding tRNA molecule. tRNA is a long chain of RNA with 3 nucleotides attached to the bottom. These nucleotides are called the anticodon, which match up to the codon being processed by the rRNA complex. Each tRNA also has an amino acid attached to the top that is released to a ribosome when its anticodon is matched up to a codon. This amino acid binds to the ribosome and forms a chain which will eventually form a protein.
Think of this as Gino has made the secret recipe book for a restaurant. The chef wants to cook a certain dish and needs to read the right recipe, copy it down for use in the kitchen. The chef carries out the role of mRNA, which takes the code from the nucleus to the ribosome. The chef then needs to bring all the ingredients to a large cooking pot, which represents the rRNA complex known as a ribosome. The ingredients represent the amino acids carried by the tRNA molecule, the chef in this example. Eventually, the finalized dish represents the completed protein.
Yeah, and then basically mutations, as I said, there's the 2 factors. So the first factor is just when your DNA is formed, there's usually a mistake that's made. So there's the point mutation, which is just a single cell base in your DNA or whatever that gets substituted. So like I mentioned earlier, could just be like, instead of ATCG, it could be AACG. It doesn't really change the whole chain much, but slightly. Mm-hmm. And then there's the frameshift mutation, as I said, which is either an addition or a deletion completely of a base in the DNA chain. So instead of being ATCG, it could completely delete the T, or it could add like 2 T's opposed to just having the 1 T, which then basically changes the whole reading of the frame altogether, which is where basically Down syndrome and autism, things like that come in humans and whatnot. So, do you kind of understand what I'm talking about?
I do.
All right.
I do get it.
Well, thank you, and I hope you enjoyed watching my video. And my cats, because they're super cute. By the way, awesome. Bye.